this kind of field of biology is the fact that you kind of have to know a little bit about a lot of different things. So it turns out, though, even though, if you're a general plant pathologist, you kind of know a lot about a little different, a lot of different things. Every pathologist has their favorite. And my favorite pathogen is a pathogen called Phytophthora infestans. And that name itself means infesting plant destroyer. And when I was going to make this slide, I kept saying my favorite pathogen, like how do I want to phrase this? And every time I said my favorite pathogen, I kept thinking of My Favorite Martian, which of course is a really older TV show, but that's what kept playing in my mind. So I was like, oh, I'll find a graphic of My Favorite Martian, and here he is. And I discovered today actually that the little things that grow out of his head look very much like these little sporangia, these little spore producing structures that Phytophthora produces. So it was like the perfect graphic. All right, so I'm going to tell you a little bit about this pathogen as we go along, a little bit about its history, the hosts that it interacts with, and the reason I'm telling you about this pathogen is because it makes zoospores, and eventually we'll get around to this whole thing of exploding zoospores that I've been researching. All right, so Phytophthora infestans causes a plant disease known as late blight. And late blight is best known for its destruction of potatoes during the Irish potato famine. And these are photos and a print from that time period. The crop losses that happened during that time period, along with the politics of the time, resulted in the death of a lot of people, at least 1 million people, and forced the immigration of another 1 million people out of Ireland. So if any of you are of Irish descent and had ancestors that came over in the mid-1800s, there's a really good chance that they came because they had to leave because of this famine that was happening. So that famine happened because of the potato crop losses, which was one of their main sources of food. And so even though this disease is often talked about in the context of history and how it changed the course of history for some people, it still is a problem and still costs upwards of $7 billion worldwide in crop losses and control measures. So it's still a problem, and people still research this disease to understand it and control it. How are we doing so far? Any questions? Okay. So now I'm going to get into a little bit more of the science of this pathogen. Phytophthora infestans is a fungus-like organism, so it's known, or it's what we call an oomycete. So oomycete, what? These are, that term actually translates as egg fungus because of the shape of some of the spores that it makes. And for a long time, scientists, pathologists, plant pathologists thought that this was that oomycetes were actually fungi, and they're also known as water molds. But they're fungus-like, and they actually belong in not the fungus kingdom, but a kingdom known as the Chromista kingdom or the Chromalveolata kingdom. So here's a couple. Here's the tree of life, one version of it. Here's fungi way down here, and can you guys see the arrow when I'm moving it around? Can you see that? Okay, yes. All right. So that's my pointer. So fungi are down here, and they're close to animals, and way up here is the Chromista kingdom, and in that Chromista kingdom is where the oomycetes live. And in this structure, you can see this round structure, and then inside of that, you see these little round brown balls. Those are oospores. That's where the term egg fungus comes from, because it looks like they make little eggs. So they are fungus-like because of their lifestyle. They do grow just like a fungus does. They have kind of filamentous hyphal growth. They absorb their nutrients just like a fungus does, and they produce spores just like true fungi do. But there are some key characteristics that pull them out of that fungal kingdom. For example, one that I think we can all relate to is this one, and that is that the cell walls of oomycetes are not made of chitin. If you have a true fungus, the cell walls are composed of chitin. That's the same stuff that insect exoskeletons are made out of. And so if you look down here and see that fungi are more closely related to animals, which includes insects, it's not too far of a stretch to say, okay, actually, they are more closely related to each other than we once thought. So oomycetes do not have chitin, though. They have cellulose and glucans. And cellulose is what we find in plants and algae. So it turns out that oomycetes, even though they look and behave very much like true fungi, are not fungi. They're actually more closely related to brown algae. They also make zoospores that have 2 flagella. Some of the true fungi make zoospores, which are just little swimming spores that we're going to talk more about, but they only have 1 flagella. And the flagella is just the thing that makes the zoospore move. So we'll see zoospores moving in a couple minutes. This was just to reiterate the fact that these guys are more closely related to brown algae than fungi. Okay, so let's, in order to talk about the zoospores, we have to kind of talk about the whole cycle and kind of see where they pop up in this cycle. So I'm going to use potatoes as my example. First of all, it's one of the more common hosts that we talk about when we talk about late blight, hence the, potato famine. It does infect other hosts that we'll talk about here in a minute. But here's a potato field, nice and healthy and happy, in Ireland actually. If these potato plants were to become infected with this disease, they would, the leaves would start to look like this. They get these dark, dead lesion areas surrounded by this kind of white, powdery looking stuff. And that white stuff is composed of a whole bunch of things we call sporangia. So I'm going to show you sporangia and also zoospores here. So let me let these graphics pull up and then I'll talk about what we're seeing. So, okay, so we start with this leaf here that has these sporangia. And if we look close under the microscope at where this white stuff is, what we will see is something that looks like this. This is a little, what we call a sporangia 4 with the sporangia on it. And these to me look like little lemon drops. Some people call them footballs, some people call them lemon drops. So this is one of those lemon drops, and under the right conditions, it will produce 6 to 8 zoospores that look like this, that have these 2 little flagella and can swim around and cause infections. So you can actually get infections 2 ways. One is that these sporangia just break off and float away and land on a leaf and cause an infection, or each one of those sporangia produces 6 to 8 zoospores that can each cause an infection. So once you get these lesions like this and you start getting spores that are being produced, they can go out and cause infections, cause more sporangia and zoospores to be made, more infections to happen. So you get this repeated cycle of infections that can happen many times over a season. And under the right conditions, this disease, which once you find out that you have it, it's really too late, which is I think why they call it late blight. Unfortunately, once you know you have it, it really is too late. Under the right conditions, that would be like kind of consistent cool or persistent cool and wet conditions, this disease will easily take down a crop of potatoes in less than 2 weeks, no problem at all, because of this rapid spore production that can happen. I should also mention it just produces, it's a prolific spore producer. So 1 little square centimeter of lesion area here will produce upwards of 20,000 of these little guys here. And if each of those 20,000 sporangia is making 6 to 8 zoospores, you're going to get a lot of disease really quickly. So that's what happens with late blight. The other thing that happens is the sporangia or the zoospores can also infect, sorry, I went the wrong way. They can also infect the tubers underground. So if you have a field that's infected and you get rid of the plants, but you leave the tubers underground and they germinate and grow the next year, you've now got another crop that's already infected before you've even started. And this is kind of what happened in Ireland. They had persistent cool, wet conditions. This new pathogen came in. They didn't really know what it was or how it worked. They saved their potatoes because that was their food. And then they planted them the next year, and some of them were infected. And so then they just perpetuated the disease. And for several seasons, they had it, and then it just wreaked major havoc. But again, it can still be a problem under the right conditions. We just know more about it now, and we know that it exists, and we know the conditions that it thrives under, and so we can work to try to control it a little bit better, mainly by applying certain fungicides or planting resistant potato varieties. So there are ways of kind of keeping it somewhat under control.
Margot, can I ask a question?
Yes, ma'am.
How do they jump from one plant to the other?
Yeah, so it will be, so if it's sporangia, it's wind or water. So it can either be raindrop or wind dispersed. The zoospores, you have to have a film of water. So if you've got persistent kind of wet conditions, like a film of water, they can swim. I don't know how far they swim, but they can live a long time. They can live several days. And so there's a couple different ways that they can spread. Good question. Thank you.
Thank you.
So potatoes are one of the hosts of this pathogen. And have any of you ever grown potatoes? Some I'm seeing yes, some no. They produce really beautiful flowers, actually. A lot of us, I think, if you don't make potatoes, if you don't produce them in your own garden, you may not see the flowers if you just get them from the grocery store, but they actually make a really pretty flower. So these are potato flowers. Tomatoes, also in this same family, also are susceptible to this disease. And petunias are also in the same family. So these are the three most common hosts for this disease. There are several others, though, and these are all, again, in the same family. So this is a huge family. This is the nightshade family, or what's known as the solanum family, or the solanaceous family, lots of different names. The potato family. There's several hosts, though, that they aren't as susceptible as these three hosts, but can get the infection to some degree. And a lot of these are weedy species and might be present, in or around fields. And so people have to be aware that they could be a source of infection for their crops. So that's another thing that a plant pathologist has to understand is, like, what's the host range of their pathogen. I stopped at mandrake because if any of you are Harry Potter fans, you may recognize that name as one of the plants from the Harry Potter series. It looked like this in the movies. It's got this folklore that goes with it because the roots actually look like this. They look like little people, and so it has conjured some folklore that these are actually little people, and when you pull them up, they scream. I don't think they actually do that, but it's still fun to talk about. All right, some other plants in this family, nightshades, and a variety of other things that some of you may be familiar with. These are just a few, but again, these three are the main hosts that we talk about when we talk about late blight, but it does actually have quite a broad host range. So of all of those species, this is the one that I have focused on. So my main focus is on Petunia late blight. So we're going to talk about that a little bit more. Before we get into Petunia late blight, the disease, and what goes on with that, I'm going to take a little detour and talk about petunias because they have a really cool history, actually. So petunias, like many of the members of that family, are native to South and Central America. And petunias in particular tend to be native to like the Argentinian area. And there are two parental species that are the source of all the hybrids that we have today, basically. We've done some other kind of hybridizations in between, but in the early to mid 1800s, the two parental species were discovered during the days when they sent out big parties of people to go out and survey and collect all the different plants and learn about them. So one group sent out by Napoleon found this plant, Petunia axillaris. It's a white flowering petunia. The other parent is Petunia integrifolia, found a few years later by another group out of Scotland. And these were both brought back to Europe in the 1800s. And in nature, they don't cross. In nature, you have the white flowering petunia that's pollinated by moths, and you have the purple flowering petunia that's pollinated by bees. And they don't cross over in nature. But if you get them back, you take them into the greenhouse and you do your own crosses, they found that you get a huge variety of shapes and sizes and colors of different petunias just from a single cross. So these were all pictures. They're all scans actually of some historical books that were in the Cornell University Library. That's where I went to graduate school, and I had the opportunity to delve into the deep guts of the library and find some of this information, which was really fun. So anyway, the first hybrids were produced in the late 1800s in Europe, and then eventually they kind of spread around the world as an ornamental flower. So in the United States, one of the first hybrids that became popular here was late 1800s, and it was these California giants. I should mention too that you can get these California giants still, and you can get those two parental species from heirloom seed companies. In fact, I grow the two parental species in the greenhouse at CMU, so you can easily get them. I have not done any of my own hybridizations. I grow them for other research purposes, but you can still get them. So anyway, from those two parents, we've gotten all the cultivars and varieties that we have now, and thousands have been developed. And in general, there's a few hundred that are kind of in circulation at any given time. So they're produced all over the world these days. Americans use probably more than this now. This is probably an old statistic, but at least 500 million petunias a year. You see them everywhere. You see them downtown. You see them in front of people's houses. That's my house. You see them everywhere, gardens. They're cold tolerant, they're drought tolerant. The newer varieties are really rain tolerant, so they can be rained on and they'll kind of pop back and look all happy and fresh again. One of my graduate school advisors used to call them the 50-mile-an-hour flower, because as you're driving along at 50 miles an hour, you still see them because they're prolific bloomers and they're bright colors, and they're just a great plant for a lot of reasons. I like them because of the research I've gotten to do with them. Okay, so speaking of that, back to late blight and why study petunias. So the reason I started studying them and continue to study them is that petunia late blight requires, well, we found as I was working in graduate school and kind of learning about this interaction between petunias and Phytophthora infestans, we found that Petunia late blight requires the same general environmental conditions as potato or tomato late blight, but they're generally much less susceptible than potatoes or tomatoes. This is an example of an inoculation that I did in a controlled growth chamber. And in the background, you see these poor, sad, very unhappy tomatoes. This is after about a week. So I infected these plants, gave them, I'll start to see infection within 2 or 3 days, and by a week's time, the tomatoes are, they're done. In the foreground, we see petunias. There's a little purple flower there, and there's a white flower here. They're hanging in there. They do get infected, but they are not looking as sad as those tomatoes, for sure. So they are much less susceptible, which is why I was working on them in grad school and have continued to work on them, just trying to understand why that might be the case. The why, we know that there's things that go on inside of the plant. They're able to kind of sense that there's an infection sooner than a tomato and a potato can, and they can fight that infection off very quickly. However, there's also things that go on the surface of the plant prior to, or that might prevent the infection from happening to begin with, and that's where the exploding zoospores come in. All right, good so far. Any other questions? Okay. All right, so now to the case of the exploding zoospores. So while I was in grad school, this is something I accidentally came across, which was kind of cool because it wasn't like, hey, we have this thing in the lab that we want you to look at that we already kind of know about. We want you to sort it out. It was like, I found it all by myself, and it was like, whoa, what's going on here? So this has been my pet project for a very long time. So I found this interesting thing going on between petunias and zoospores of Phytophthora infestans. What I found, so I'm going to kind of let the cat out of the bag, which you guys, based on this title, you already know there are exploding zoospores. I found that the petunias were killing the zoospores, which is cool on the one hand, but the day that I discovered it, it was like I was in a total panic because I needed those zoospores because I was doing experiments. I was like, no, 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 don't do that. Experiment went fine. I got that done, and then I was able to go back and try to figure out what was going on. So I made this graphic many years ago, but I keep using it because it took me a long time to make it using Photoshop. I'm not very good at Photoshop, and I'm still not very good at Photoshop. So this is a killer tomato face that I put on the top of a petunia and call it a killer petunia. So that's my graphic that will live with me forever. But I always bring this up because I think this is funny. There's in the process of trying to find a graphic that was a killer petunia, I found a Canadian music group called the Angry Petunias. They're from like the 80s, 90s. Their music is terrible. I wouldn't recommend it, but they are really funny. If I had more time and I was in a class, sometimes I share the music with my students, but I won't torture you with that. Okay, so here's how it went down. All right, so like I said, I was in the process of doing an experiment and things kind of went wrong. Normally I would take infected tomatoes. So in the background here, I've got an infected tomato, and I would just kind of wash those sporangia off into water, and I'll show you that process in a second. Let me just do that now. So I would, in order to infect a plant, I have to get spores to put on the plant. So I would take spores from an already infected plant, wash those spores off into water, put that water that now has sporangia and zoospores into the refrigerator, and that stimulates the sporangia to produce these zoospores. So normally I would do this with infected tomato leaves. Everything goes as planned. I get happy zoospores. They're swimming around in there as happy as can be. Let me show you what happy zoospores look like before we talk about exploding zoospores. So I'm going to come back to this slide in a second, but I want to show you some videos. So first, this is a close-up of a zoospore under the electron microscope. It's got two little flagella here, and those flagella whip and spin around and make the zoospore swim. This video up here on the top I'm going to show you first is going to show you actual moving zoospores. This is a video from a lab in Berkeley. And this is a different Phytophthora. This is Phytophthora ramorum, which causes an infection on oak trees. So here, all right, so under the right environmental conditions, it's going to produce these little sporangia will produce infectious zoospores like I was talking about. So cool, wet conditions. So you get these little zoospores, and once they're ready to go, they have to come out of these sporangia. Okay, so they have to emerge out of the sporangia. So it's like watching birth. These little zoospores are being birthed. So they come out of the sporangia, and they get their flagella going, and then they swim off and they cause infections. So those are happy zoospores. That's what the zoospore wants. It's not what the plant wants, but that's what zoospores want. They want to be happy. You get enough of these in a solution, and this is just kind of a fun little aside here, but this is from my lab, and if you get enough of these, so this is looking down on the top of a beaker of water that has zoospores in it, and if you get enough of them in the water, they start to do this cool aggregation thing. So this little moving cloud that you see right here is millions of zoospores in the water. They like to be at the surface of the water. So they come to the surface and they kind of hang out and bump into each other, and then you get too many and some of them fall down, and then you get more coming up, and so they just kind of do this really cool aggregation thing. So that's an example of happy zoospores, lots of swimming zoospores, and that's what I would normally see. Sorry, get this. Yep. That's what I would normally see on any given day if I was just using my tomatoes as my source of spores. But on this day, like I said, my tomatoes were in trouble. I wasn't getting enough spores, and I threw in some petunias because I had some infected petunias. So here's some petunias here that have some spores. So I threw them all together and did the usual. Threw them in the fridge, checked on them in 30 minutes, and what I saw was zoospores exploding, and that's when I kind of panicked. But as I said, got that taken care of, and then came back on another day and tried again, focusing on the exploding zoospore problem and what might be going on. So I started to kind of sort things out and try to figure out where was this problem coming from. So I initially actually thought that it was the ooey gooey tomatoes that I had that were maybe causing the problem. Maybe there was some junk in there that was affecting the zoospores. So I did another round with the ooey gooey tomatoes, and that wasn't causing the trouble. But if I took just the petunias all by themselves and rinsed them off into water and then took them through this process and looked for zoospores in 30 minutes, they were exploding. So it seemed to be something coming from the petunias. The question then was it something that was causing the zoospores to be kind of like not formed correctly during the process of development on a petunia, or was it something that was in the solution around the zoospores coming from the petunias? So in order to kind of answer that question, I took petunias with the sporangia, rinsed them off into the water, and then filtered them, got them away from that petunia water and into some clean water, and then watched to see what happened. And in that case, the zoospores were fine. So I was left with these happy zoospores that had come from the petunias, but they were separated from the icky petunia water. So now I had this icky petunia water that maybe could be part of the answer. So the next question was, well, what happens if I take happy zoospores and I add some of that icky petunia water to those zoospores? Will they explode? Well, let's see. So this is a video from my lab also to see what happens. So this is, and this goes really fast, so I'm going to show it a couple of times. I'll pause it also. Okay, so initially, first off, remember that, so that aggregation that I just showed you, that kind of moving cloud of zoospores, if you get down into it and look at it under the microscope, this is what it looks like. It's just millions and millions of zoospores swimming around, very happy. Now you're going to see some commotion. That commotion was me adding some of that petunia water to the zoospore. So I've just added some of that petunia water. And then you're going to see some more commotion as I try to adjust the microscope to get in close to the zoospores to see what's going on. All right, so we zoom in closer, try to get it focused quickly. By the time we get it focused, look at these two zoospores. Right? There's one that popped and boom, there goes a second one. So that's all in real time. So let me play it up to the point where those zoospores are getting ready to pop again. So I've added it, and the time it takes me to get the microscope adjusted so that we can see what's going on, we actually already have zoospores that have exploded down here and here and here, and then these two are getting ready to explode. So it happens really quickly. All right, boom, boom. All right, did everybody see those explode? Okay, so that was what was happening, and that's why I was panicking, because I needed those zoospores to cause, to do my experiments. But now that I had gotten my experiment done, I could kind of focus on what was making this happen. So from here, once I sorted out the fact that it had something specifically to do with petunias, fast forward a bit to what we now know and what we think is happening. So what we now know is that petunias produce these sugary compounds called sucrose esters on their leaves. How many of you have, or I guess I should say, have any of you ever gardened with petunias? Have you ever noticed that they're sticky? If you haven't, the next time you handle petunias, notice how sticky their leaves are. And the reason they're sticky is because they have these sucrose esters. It's the same sucrose it's sugar. It's the sucrose from sugar. So that's why they're sticky. They're making these sugary substances on their leaf surface. I'm going to throw a little chemistry at you, but don't panic. I'll walk you through it. So this is a sucrose ester molecule. So here we have sucrose. This is what sucrose looks like at the chemical, molecular, anatomical level. They're called sucrose esters because we have a sucrose, and then we can have a bunch of things that can attach to that sucrose via a bond that we call an ester bond. So that's where the sucrose ester comes into play. Now, what's attached to that sucrose can vary a little bit, but we know that in petunias, a lot of times what's attached to this sucrose is one or more fatty acids. So this thing here is meant to represent this fatty acid tail. And it's that structure, that fatty acid tail, that we think is really responsible for the explosion of the zoospores. So what we think is going on is that these fatty acid tails, they're fats, will integrate into and, well, basically, you know that oil doesn't like water, but oil likes oil, right? So if you've got oil and water, all the oil likes to go together and all the water likes to stay out here. So these are fats, these are oils. The membrane of a zoospore is also made up of fats. It's made up of phospholipids. So we've got lipids and fats. And so these things like to interact with each other. That fatty acid tail can get into the membrane of the zoospore and start poking holes. And since these zoospores are in water, that means water can get in, and it can get in faster than the zoospores can get rid of it. And so we think this is probably the mechanism by which petunias are causing the zoospores to explode. They're producing all these sucrose esters on their surface. When the zoospores start swimming around trying to cause infections, they're actually exploding before they can cause an infection. Some of them do get through and will cause infections, but once they get through, then the petunias turn on basically their immune system and shut that down pretty quickly. So just to give you a graphic of all the things I was trying to do with my hands. Here's your sucrose ester again. Here's that fatty acid tail. Likes to integrate in and interact with the fats of the membrane. So this is a portion of that zoospore membrane. When it does that, if you get enough of them into one place, those sucrose esters like to hang out with each other. And they can actually potentially form holes, like pores, within the zoospore membrane. And this is where the water would be coming in and the zoospore can't get it out quickly enough, and then it pops. So that's what we think is happening. And that's what we're focused on kind of now, is understanding that mechanism a little bit more. So I did not do all of this by myself. I started it in grad school, but since then I have had 10, there might actually be 11 on this slide now, at least 10 undergraduates that have worked with me over the years on various parts of this project. So I have to thank them for their help, and that's part of my job too, is to bring students into the lab and show them what it's like to work in a lab instead of just sitting in the classroom. All right, I'm going to shift gears here. Are there questions that I can answer before we do that? Okay, so this is where we're going to shift to the flesh-eating fungi. So I will tell you that, as I said at the beginning, there's nothing too super gross here. The flesh-eating fungi that we're talking about are actually come into play after someone has died. All right, I'm not talking about flesh-eating fungi that would attack you today. So how did I get here, right? So I'm a plant biologist, and I never in a million years thought I would be looking at fungi that consume human flesh. But basically it comes down to the fact that other people assume that I must know something, and I like all things biology. So this is that Jill of all trades that kind of comes out in me. And also this particular project, to be fair, excuse me, came to me because of my plant pathology background. I have the opportunity to teach the mycology course at CMU. So mycology is the study of fungi and fungal biology. So because I teach this class, this led people around me to think that I must know something about mushrooms and molds. And I do know, after teaching this class for several years, I do know a little something. So that's why this project, when it came up, was intriguing. So first of all, these are not flesh-eating fungi. These are just mushrooms that were found on the Grand Mesa by one of my students during one semester, one particularly really good year when it was actually wet. This year's not going to be a great mushroom year. It's too dry. But anyway, this is a really beautiful glamour shot of the diversity of fungi that can be found just up on the mesa. Okay, so this class, though, is really the reason why I got into this current project. And that is because I had some students a few semesters ago that were taking mycology, and they were also interns out at what we call the body farm. And part of what students do in this class is grow molds just from around. And you can put a petri dish out and you can catch so many molds just from the air. But a few years ago, so the body farm, the actual real term for it is the Forensic Investigation Research Station, but a lot of people just call it the body farm. So the director is Dr. Melissa Connor, and she had brought to the attention of the students that were interning there and also taking my mycology course, the fact that there were some cadavers that they have purposely left out to watch the decomposition process over multiple years under the conditions that we live in here, which is a high desert environment, very dry, lots of sun, and just to understand what the process is. So she had pointed out to the students that some of them had kind of a black crust or kind of a black mold, she thought, on them. So this is that black crust. And if I crop this picture down even more, and I didn't tell you what this was, if you're familiar at all with kind of the desert soil around here, you might look at that and be like, oh, it's just desert soil, it's just desert crust. But that's actually skin. It's a mummified skin of a cadaver that's been out in our environment for, at that point, at least 3 years. Now we're coming up on 5 years. So these are the two pictures of skin and bones that I will show you. That's as much as you're going to see of the tissue, because this is what we're sampling from. This is bone here, and there's some skin around it. It's just very dry, looks very crusty, just like soil crust. But Melissa was curious, and so she had the students collect some samples and bring them into class, and we plated them out. We started to see some interesting things, and since then we've done some more work. So this is still a new project, and we're still learning what's going on here, but I'll share with you what we know so far. So first of all, why even study fungi and human remains? So first of all, decomposition happens in phases. So anything that's decomposing, whether it's an animal or a person, it's going to happen in phases. And understanding those phases helps determine what's called the postmortem interval, which is also known as the PMI. And understanding the PMI helps with crime scene investigations. How long has a body been there? So the integration of understanding how fungi might play a role is called forensic mycology. And I'm going to take a minute to shout out to my daughter. My 13-year-old helped me with this graphic because I needed something that had forensic science and mycology. And I found two graphics. One was one that just said forensic science and one that had a mushroom spore print. This original graphic had a fingerprint, so she erased the fingerprint and put the spore print on there for me, and voila, I have a forensic mycology graphic. So she made this graphic here, so I just wanted to shout out to her because that was a huge help to me. She does know how to use all those graphic design things, which is super awesome. Okay, so studies in the area of forensic mycology have suggested that the presence of certain fungal species help determine that postmortem interval. So if you watch the decomposition process, maybe if you don't know how long a body has been there and you take some samples from it, you may be able to determine based on what organisms are there, how long it has been there. There's also a whole area of forensic entomology that goes into this because there are insects that are involved in this process as well. And there's a ton of bacteria. So there's a lot going on there. This is just one part of that. As I mentioned, we're in a high desert environment. Environment plays a huge role in how that process plays out. The fact that there are cadavers at the body farm right now that are still pretty much fully intact, I think is somewhat unique to the fact that they are here. If they were someplace where it was more wet, warmer and wetter than we are here, then they would decompose much quicker, and you may not see some of the fungi that we are seeing at this stage. So because of the location of furs, it gives us a unique opportunity to learn some things about a unique environment and how this process works in that unique environment. Okay, so I'm going to quickly just run through the results that we have so far, and this is a lot of pictures of fungi, well, molds and spores and things, so it's not super interesting unless you like looking at molds under the microscope, which I do, so you have to pretend that you like it today. So here's, these are some plates. So we took some samples from some skin and some bone and we put it onto plates so that the fungi could grow. And honestly, I was expecting to see so many different fungi that we wouldn't be able to sort them out. And it would just be like, wow, okay, there's a lot there. But it turns out that, so first of all, this is kind of typical growth after just 4 days or so of just some small samples that we took from those sites. And surprisingly, every time we've taken samples over the last several years, we get 3 fungi that we consistently find every single time. Now, there are probably others there that we can explore a little bit, but every time, whether it's from skin or bone, we're usually getting at least 3 that consistently show up every single time. So that's what we've focused on, is trying to figure out what those are. And if they're, what are they? Are they like flesh? Are they actually flesh-eating fungi? Are they unique, or what are they? So the 3 that we tend to see are these. One is this kind of shiny black fungus, and you see a lot of that black fungus in this particular plate. Another one is this kind of gray, brown, greenish mold that grows, and you see it in multiple places throughout here. And again, I admit there are some others in these plates, but these are the 3 that we see consistently every time. The third one we see are these kind of diffuse brown colonies. So there's one there, and there's one there, and there's actually one up here, and there's one kind of buried in here. So this is a mixed plate of different fungi. So from here, once we kind of let things grow out a little bit, and actually before it gets to this point, these are a little overgrown, we would take samples of each of these fungi and put them onto clean plates, fresh plates, so that they can grow into a pure culture all by themselves, so that we can, you know, look at them and not worry about, you know, having a whole bunch of other things in that same plate. So we take these subcultures and we put them into pure, put these fungi into pure cultures, and that allows us to look at them, look at their spores, and also to get DNA to extract in order to kind of confirm our identification that we might be able to do using their spores. We can confirm that using DNA sequencing. So what we've found so far, that little black shiny guy looks like this in pure culture. Kind of has this black center, white edges, smells very yeasty. This is what it looks like under the microscope. Lots of black spores, and towards the tip where the white is here, this looks more, these little guys right here look more like you would think when you think of, like when you smell yeast and you look under the microscope, you expect to see a lot of little small white clearish spores, not black spores. But it turns out that there are in fact black yeast. And the morphological characteristics that we saw under the microscope did suggest some type of black yeast. The DNA sequencing that we did to follow up on that did identify it as a black yeast in the genus Areobasidium, which is a yeast that's everywhere. It happens to be a salt-tolerant yeast, which is kind of interesting because the soils that we have around here are very salty, alkaline soils. So the fact that it's around, you know, maybe it's just in the environment, but it has also been isolated from other desert environments. So it's very heat tolerant, it's very salt tolerant, which is kind of interesting. The dark color of it suggests that it's this genus here, Melanogenum, because it produces melanin. It's the same melanin that we produce in our skin, fungi produce in their spores often. That's one. The second one was that kind of gray or brown, green, multiple kind of colors really. You look at it under the microscope and it makes these funky looking little spores that have all sorts of kind of different shapes and cross walls and various things. Based on what those spores looked like, I thought it was one of 2 fungi. I actually really thought it was this first one, this Eulocladium, which is a really common mold, again, that you can catch easily just by putting a plate out on your countertop. It's everywhere. The alternaria is what the DNA sequences strongly suggested. It's also everywhere. So both of these are ubiquitous fungi. They're just kind of all over the place. The third one, that kind of diffuse brown fungi, this is what it looks like in pure culture, a little bigger colony than those initial ones I pointed out. When you get close on it, these make these little flask-shaped structures called pycnidia, and there are little tiny millions of little spores inside of these little flasks. And when you flood these plates with water, the spores just kind of ooze out all over the place. So this is based on that morphology. I thought it was this fungus known as Foma, and the DNA sequencing strongly suggested Foma as a possibility. Foma is another just fungus that's just everywhere. It's just ubiquitous. It's kind of wherever there's stuff, there's Foma. So all three of the fungi that we've been isolating are not really specialty fungi. They're not, I mean, I called them flesh-eating fungi because that was my hook to get you in, but they're not really flesh-eating fungi. They just chew on whatever they can find. So it's pretty likely that they're just in the environment and they happen upon a cadaver and they're like, all right, I'll chew on this for a while. But maybe they don't chew on cadavers until late in the game. We don't know. That's one thing we don't know. So I've got kind of a list of like what's next for this project. Before I get too far with that, though, I need to make sure that I do give credit where credit is due, and that is for the sequencing. While I did do the morphological work, I really had very little to do with all the sequencing work. That was due entirely to the hard work of my most recent research student, Selena Tucker, who's off to graduate school, and my colleague, Zainab Asay, who's on here today. I have to say thank you very much to both of them. The sequencing wouldn't have been done without them because that's outside of my wheelhouse. I'm perfectly happy to be in the lab looking under the microscope at spores all day long, but if you ask me to isolate DNA, I can't do it. So thanks to Zainab and Selena for that. So what's next for this project? Are there other fungi of significance that we've missed, perhaps? Do our findings, I should say findings, sorry, do our findings match what others have found? If others have found, so this whole what's going on very late in the decomposition stages is something that, as far as I know, isn't something that's researched super heavily. So it's kind of interesting to learn. Also, what fungi are present early in the decomposition process, which is something that more people have looked at. And are the three that we have identified, are they part of those earlier stages, or do they just come in later? We have a little bit of preliminary data about what's present during the early stages, and that's why I've got these two pictures. This is an orange with multiple species of penicillium, another very common mold. And this is mucor, which is another common mold. Both of these are kind of under the umbrella of common bread molds. But they kind of chew on whatever they can find also. And so they've been documented. We have isolated them from kind of earlier stages of decomposition, and so have other labs. So again, the whole reason we look at this is just to kind of understand what's that process of decomposition, and are there markers of certain species of fungi or bacteria or insects that you can tag as, okay, we're seeing this group of organisms, so that means that our timeline is about here. And that helps with crime scene investigations. So that is how that project is going. And the research, of course, will continue from here. And I'm finally done talking. Can I answer any